Dr. Madangi Ganapati has obtained her MSc in Genetics and Plant Breeding from Kerala University and PhD from School of Biotechnology, Madurai Kamaraj University. Having a passion for gaining knowledge and for instilling curiosity in fertile minds, she has been active in the field of teaching and research for more than 15 years. Her current research interest includes recombinant protein expression in plants, transgenic plant development and manipulation using molecular techniques, and analysis of changes in basic gene regulatory pathways during transgene expressions, nutraceuticals, and the role in alleviating certain chronic human diseases. She has a number of publications in peer-reviewed international and national journals.
Welcome to the lecture series on food microbiology. Today we are going to talk about an entirely new subject which is called as natural antimicrobials. These natural antimicrobials are substances which are synthesized by either microbes or by some from natural sources as a sense of protection, in the sense that if it is secreted by a bacterium, then this is a protection method for that bacterium to evade or not to allow other strains of bacteria from colonizing the same product, or maybe if you are culturing it in the lab, against using the nutrition in the same plate. Among them, one of the chief compound we talk about, which is synthesized by the bacteria, we call them as bacteriocins. By definition, bacteriocin is a protein which is produced by bacteria of one strain, and normally this is active against those of a closely related strain, meaning if a bacteria synthesizes a particular type of protein, then this is going to inhibit against a similar or closely related species or a genus which will compete with it for the same carbon source or the same energy source. So that, see, if you have two or more strains which utilize the same carbon or same energy source, you have a competition for the substrates, which will prevent all of them from proliferating well. So during the course of evolution, so the bacteria, to protect itself, to proliferate, synthesizes these bacteriocins. Potentially, these bacteriocins can also be taken as an alternative to the traditional antibiotics, because these are, as I said, they are proteins or polypeptides. They have high potency because they will completely inhibit the closely related species or closely related strains, and they have low toxicity, which is very important, and they can be produced in situ by probiotics and can be bioengineered. Now, this sentence needs to be explained. Probiotics is a term which all of you might have heard about. Probiotics are what we call as the beneficial or the good microbiota. For example, you have a gut microbiome. The microbiota there help you in the digestion of food, in the absorption of food. They are the ones which synthesize enzymes which break down most of the complex stuff which we consume as a food and then provide nutrients for us. So, these peptides, so see, you have a microbial colony inside. So, when you have such a colony, you require the proliferation of a balanced type of organism, a balanced what we call ratio there. So, this can be maintained by these organisms themselves by producing these bacteriocins. Once we know what are these and which are the bacteriocins which are beneficial, we can also, using genetic engineering techniques, create strains which can synthesize these bacteriocins and provide them. So, that is what we mean, that which can be produced in situ by probiotics and can also be bioengineered. And they can be broad spectrum or narrow spectrum, meaning they will inhibit or be active against a very narrow amount, that is, number of strains with which it can inhibit would be very less, or it has a broad range where a large amount of organisms can be inhibited. So, this is the general term called as bacteriocins. Among this, you have very well known ones like your nisin, subtilin, niacin, etc. We are going to discuss about a couple of them, mostly with applications in the food. Nisin. This nisin is a low molecular weight polypeptide produced by the dairy starter culture Lactococcus lactis subspecies lactis, and this is a broad spectrum bacteriocin. So, the source organism is Lactococcus lactis. Now, what does it do? if you talk about fermentation, if you want to produce curd or yogurt or cheese in the dairy industry, you require a starter culture of a particular microorganism. And this starter culture, normally we use a pure culture, meaning the same genus, same species. Each industry or each industry, for example, if we talk about India, you have Amul, you have dairy, so many different types of cheese varieties are available. So for each of them, if you look into the taste or the flavor, the product taste and the flavor differs. This flavor difference is ultimately caused because of the specific starter culture which they use for the fermentation. Normal milk or normal dairy products do have this, but it will not be homogeneous. The type of Lactococcus species which inhabit the normal would be different. You have a heterogeneous mass. But here, to get the commercial grade nisin, normally we use the starter culture Lactococcus lactis, and this gives rise to the nisin. And this nisin, which is thus produced by Lactococcus species, is active against a wide range of gram-positive bacteria. This is used as a food preservative as it can maintain or extend the shelf life of heat-treated fruits. One aspect is preservation. The second aspect is it can also reduce. The moment you say preservation, preservation is because you are going to inhibit the spoilage microorganisms. And what are the spoilage microorganisms? Again, the microbes which are pathogens or which are spoilage. So if they get inhibited, it automatically means that the shelf life of the food gets increased. You have variations of this nisin. The nisaplin, which is otherwise called as nisin A, this was the first commercial extract which was developed between 1962 and 1965 from the naturally occurring lactococci which produce nisin. This is normally seen. You have what you call as for industry grade production of nisin, you use a starter culture which is pure, but normal milk also has nisin producing lactococci. So even in normally sold milk and cheese, you do have some amount of nisin. Nisin is a primary metabolite produced by a process involving ribosomal transcription and translation, meaning this is a normal polypeptide or a protein synthesized by the organism during its normal protein synthetic machinery. That is, the gene for this dwells in the genome of the bacterium. When its metabolism is active, the gene gets activated, it is transcribed by the ribosome, and it is utilized by this protein produced is utilized by the organism as a natural process. So the basic transcription translation mechanism of the bacterium produces nisin, which is a primary metabolite. It is not therapeutically used either in human or veterinary medicines. It is not used as an animal feed nor for growth promotion. This is actually very important. If a compound has to be used as a food preservative, then its therapeutical application has to be very less or absolutely zero, both not only for humans but also in veterinary medication. Most of you would have heard about antibiotic resistance, right? Now, nisin is a bacterial product, and antibiotic resistance is something which the bacteria gains when you use an antibiotic constantly against it. Suppose, and I said nisin earlier has a potential alternative for an antibiotic. So it can be utilized in the place of an antibiotic. Now, how do you gain resistance? Because you indiscriminately keep on eating antibiotics, as a result of which resistant bacteria pile up, they become. So when you have such resistant microbes, then this nisin, what antibiotic which is supposed to treat you and cure you will not work. That is the basic mechanism of antibiotic resistance. So anything food grade which has to be used, it should be something which is not therapeutically either in human or veterinary medicine, because if the food is preserved using an antibiotic or using a potential alternative which is already prescribed for human or veterinary medicine, there is every possibility that the organism, the spoilage organism, would have already got resistance. which means that this is not going to be active, this will not work. Hence, this class that nisin is not used therapeutically, or that it is used even as an animal feed additive, because meat and meat products are we get from the animals. If the animal is, if the nisin is applied to the animal either as an additive or as a therapeutic veterinary medicine, then you could have the development of lactococci which are resistant to nisin. So, this becomes very important under the scenario. If you see the structure of nisin, it is a polypeptide, as I said. So, it has 34 amino acid residues. What is more peculiar to this polypeptide is there are certain uncommon or not very rare amino acids which happen in this polypeptide, that is the lanthionine, represented as LAN, the methylanthionine, the didehydroalanine, and the dihydroaminobutyric acid. The four which I have said before, they are very rare. They do not occur normally in many other proteins. And the presence of these four give a specific advantage to nisin. And if you see the internal structure, for example, if we use a common nisin A, there are disulfide bonds or bridges inside the internal structure with 5 internal rings. And you have a natural variant nisin Z, which differs by the substitution of a histidine for asparagine at position 27, meaning as I said, this is the 34 amino acid chain. So, the nisin A has a particular stretch as amino acid A, amino acid 1, 2, 3, 4, the 34 amino acid stretch. In that particular stretch, the variation what you see in the nisin Z is that at the 27th position, instead of asparagine which is seen in nisin A, you are going to have histidine. The amino acids lanthionine, methylanthionine, didehydroalanine, and the dihydroaminobutyric acid are normally introduced into the by post-translational modification of the precursor polypeptide, meaning you have the polypeptide getting synthesized during coupled transcription translation, and after that, as a post-translational modification, you are going to insert these unusual amino acids, and finally, after this addition, then this will get converted into the final peptide, and the basic sources for the production of these unusual amino acids is from serine and threonine, and also with an enzyme catalyzed addition of cysteine residues to the dihydroamino acid result in a multiple thioetherne. So, it means that during the primary metabolism, when nisin is getting synthesized, you have a series of steps for the insertion of these unusual amino acids and to give you the ultimate fine structure of the natural nisin which is synthesized. The net charge of nisin is pH dependent. It is amphipathic in character. The N terminal contains several hydrophobic residues. The C terminal is hydrophilic. Once it was established that nisin can be used as a preservative, then, and if we are going to use it as a food preservative, then you have to have a comparison with the synthetic preservatives which are normally used. So, and also its action with the normal natural emulsifiers which we add during food processing. So, scientists tried to compare that. So, what they found was that tween 80, which is a normal common non-ionic food emulsifier, and beta casein, which is again a food emulsion stabilizer. Okay? So, this nisin is comparable to both, which means that it can be used. But again, the effect of nisin is highly dependent on the pH as well as what is the concentration which you are going to use for the particular type of food concerned. So, it is not only the effectivity, but the type of food, the pH of the food, where it is, how it is during the process and final stage, as well as how much concentration of the nisin are you going to add to the final product. That also will decide whether this is going to act as a preservative and increase the shelf life of the product concerned. And in the food industry, nisin is obtained from culturing Lactococcus lactis using natural substrates, meaning the substrate is going to be milk or dextrose. There is no chemical synthesis. What we are using in the food industry is the natural nisin, which is obtained from Lactococcus lactis as it grows on milk or dextrose during its primary metabolism. The second question which comes is, does it kill only actively dividing cells, or is it also capable of killing the spores which are dormant? As we have already seen, bacterial spores are very difficult to control, very difficult to kill unless it starts germinating. Once germinating, it will not go back to dormancy. Then at that particular condition, we can kill them. And it is seen that nisin is active against both bacterial cells and the heat resistant spores. Hence, pasteurized as well as heat treated foods, even which are not fully sterilized, can be preserved using nisin as a preservative. But lactic acid bacteria are nisin sensitive. So, if the organisms which cause spoilage of low pH foods such as salad dressings, sauces or alcoholic beverages like wine, beer, that are even not heat processed can be used. And it is also equally effective against the food associated pathogen Listeria, then the meat spoilage organism Brocotrix. All these will get killed. Net result, one, if the pathogen is killed, you are going to escape from food poisoning. Two, if the spoilage organism is killed, naturally the shelf life of the food gets increased. And in all these cases, nisin, because it is natural, would be extremely acceptable. Why specifically it targets gram positive and why not gram negative? That is the second question which comes. Gram negative bacteria are resistant to nisin because the antimicrobial is unable to penetrate in the complex cell wall to reach the site of action, which is the cytoplasmic membrane, which means that gram negatives, as you know, they have the cell wall, and ultimately inside the cell wall you have inside it is a cytoplasmic membrane. So, if the cell wall is breached, then the nisin can go into the cytoplasmic membrane, disrupt the membrane and then break open the bacterial cell. But here in the case of gram negatives, the cell wall forms a big layer of protection and nisin cannot penetrate it unless the cell wall is damaged in some way or the other. Because of this, the gram negative bacteria are resistant to nisin, and they are also able to penetrate yeast cell walls. Therefore, nisin can be used in food and beverages prescribed by yeast fermentation. Now, when products are fermented using yeast, you want the yeast to grow, you don't want it to get destroyed, but whereas you don't want the other microbes to grow. So, in products which are produced using yeast fermentation, nisin can be used because potentially yeast will be there, but the other cells will be destroyed. Nisin works in a concentration dependent manner, hence heavily contaminated food will require higher nisin levels for adequate preservation. So, if you consider the structure and the nature of nisin, as I have been telling, it is dependent on pH, it is dependent on concentration. So, the activity you will have to, if nisin has to be used as a preservative for a particular type of food, you will first have to check the pH of the particular food concerned, find out whether it is a pH where the nisin can be active, that is number one. Two, what is the concentration of nisin which you are going to apply, because that is also very, very important. At very low concentration, sometimes it will not work at all, potentially, especially if the normal microbiota which is present on the food is very high and nisin concentration is actually very low, then preservation obviously will not happen. So, concentration as well as pH are two major factors which are going to affect the activity of nisin. How does this nisin work, the mode of action? Nisin acts on the energized vegetative cells and membrane vesicles by inserting into the membrane, forming pores and dissipating the proton motive force. This inhibits the uptake of amino acids and promotes rapid efflux of the smaller metabolites, ions or cytoplasmic solutes such as amino acids and nucleotides. Now, for the normal functioning of any bacterial cell, it requires transport of ions and molecules across the membrane, both into and out the cell membrane. And as you know, you have specific transporters which can transport, because the cytoplasmic membrane is selectively permeable, it will not randomly allow anything to cross. So, you have specific transporter systems which help for the transport of these ions, amino acids and nucleotides. Now, when this and this most cases This transport is because of a proton-matter force by the membrane potential. So when there is a disruption of this membrane potential, the uptake or uptake of these ions and amino acids would be disrupted, as a result of which you can have a lysis of the cell membrane, disruption of the cell membrane, causing bacterial death. When nisin is used in combination with a chelating agent like disodium ethylene diamine tetraestic acid EDTA, this can be effective against gram-negative bacteria like Salmonella, E. coli and other members of the Enterobacteria family. Because of the peculiarity of the cell wall structure of gram-negative bacteria, we saw earlier that nisin cannot permeate the cell wall and go into the cytoplasmic membrane, cause a disruption. But when EDTA or any other chelating agent is added along with nisin, the combined activity of both can disrupt the cytoplasmic membrane, thereby rendering nisin effective. And especially in food products, this is important because Salmonella, the specific types of E. coli and the other Enterobacteriaceae family normally are foodborne pathogens, potentially causing many gastrointestinal upsets and diseases. The chelating agents actually remove the divalent cations from the gram-negative walls. So the phospholipids and the lipoproteins would be removed or would be released by increasing the cell wall permeability. So when you have a chelating agent like your EDTA combined along with the nisin, the chelating agents will remove the divalent cations. So when the divalent cations which hold the phospholipids are removed, the phospholipids and the lipoproteins get released. So what you are going to have is pores or openings on the cell membrane, cell wall become more porous, so you are going to have a total disruption of the transport. Nisin works better in liquid or homogeneous foods compared to solid or heterogeneous products because the bacteriocin can be better and more evenly distributed throughout the food matrix. This is actually self-explanatory because when you add nisin, and since it is pH dependent and concentration dependent, for it to be very effective, this has to be spread uniformly throughout the food. And as you know, spreading uniformly would be easier in the case of liquid or homogeneous foods, whereas it is difficult in the case of solid. Hence, nisin looks better, it is the better preservative for liquid and homogeneous foods. Certain food additives should be avoided in foods preserved with nisin, meaning there are additives or preservatives which are normally used, the chemical preservatives, which can inhibit or stop the activity of nisin. So if you are going to add nisin as a food preservative, then the other chemical preservatives should not be added because it will be counter-effective. Nisin is degraded in the presence of sodium metabisulfite. This is a normal agent which is used in food as antioxidant, bleach, antimicrobial, and titanium dioxide which is used as a flour whitener. So if you are going to use nisin for preservation, then the food should not have either sodium metabisulfite or titanium dioxide. And also anything containing the polyvalent cations like calcium, magnesium, etc. would also reduce the efficacy of especially nisin Z. Let us see applications of this bacteriocin nisin. Basically in the dairy industry, especially processed cheese and cheese spreads. The second is in the milk and the other dairy products. Nisin can protect against temperature, abuse and extend the shelf life. Normally we pasteurize the milk or ultra high temperature pasteurization is done. So during this case, especially when the nisin is added at a concentration of 1 microgram per ml, and then you have increased shelf life. This is seen, this is very well observed as a preservation. This is how the normal commercially obtained nisin powder looks like. The second application is preservation of canned vegetables like canned beans in tomato sauce or tomato juice. Here we add nisin can be added as a liquid solution by combining this with a small amount of volume of canning brine and then thoroughly mixing it to the bulk fluid. So what we do is we are going to talk about canning beans, for example, in tomato sauce. So what you take and definitely in such a scenario you will be adding brine. So, small amount or required concentration of brine, remember nisin is concentration dependent. So, for this particular required volume, you add this much microgram per ml concentration of nisin, mix it in the brine and then mix that brine to the bulk of the stuff which you want to preserve. That way you will have a uniform spreading of the nisin, which in turn will preserve the food content. This is another example for the nisin which is available, commercially available in the market. This control of bacterial growth and increase in shelf life by using nisin in the canned vegetable, especially it has been reported for asparagus. And all of you know that when you label a food component, when you label especially a food product, the labeling should contain the additives, the preservatives, etc., which have been used for the particular component. So, normally, and we represent it as an E number. This E number of nisin is E234 as a food additive, and this is marketed by the Handaria as nisin Z, as a vegetal nisin Z by this particular, as a commercial product which is available. The second bacteriocin which we are going to talk about is natamycin. This is an antibiotic. It is also known by the names pymerazine, tenasectin and miprozine. This is a polyene, and this antibiotic is very effective against yeast, mold, but it does not work against bacteria. So, if you are trying to preserve, if you are going to compound for preservation, or if you are going to inhibit yeast and mold, then natamycin can be used, but not against bacteria. This natamycin is normally used as an antifungal medication, especially for fungal infections around the eye. The name natamycin came because it was isolated from Streptomyces natalensis. Now, why does this come as an application in food is, its limited use as a clinical agent. It is normally not used as a feed additive. And how does this work? Like any other polyene antibiotic, this natamycin will bind to the membrane sterols. So, when the membrane sterols are bound, we are talking about a ligand receptor binding. So, when the membrane sterols are bound, you will have a distortion, a change in the selective membrane permeability. This selective membrane permeability in turn will change the membrane potential, affect or disrupt ionic transport or transport of amino acids into the uptake of nutrients into and out of the cell, thereby causing disruption, ultimately lysing the bacterial cell. Now, fungal cells have sterols, but bacterial membrane, as you know, do not have sterols. If you remember basic structure of a fungal cell wall, you have ergosterols there. So, these sterols are going to be the binding sites for the natamycin, whereas bacteria, they do not have membrane sterols. That is why this cannot be used as a control against bacteria, but can be used effectively against fungi. And the second question which comes is, if you are going to be, since this is an antibiotic, then will fungi gain resistance to this? So far we have seen that very low level of resistance among fungi has been seen against this particular natamycin, and also it does not have any cross resistance among any other antifungal polyenes. Now, why this is important is, normally when we are talking about an antibiotic against a bacterium, in most of the cases antibiotic resistance crops up because the bacteria have plasmids which have the antibiotic resistant gene, and these plasmids can be transferred between a bacterium having it and a bacterium lacking it. So, the DNA transfer is very common by conjugation. So many other methods are there which is very common among bacteria, but in fungi such DNA transfer normally will not occur. Will not occur, but it is not as prevalent as it is in bacteria. Hence, development of resistance to natamycin is actually very less when you compare to the development of antibiotic resistance by bacteria against a particular antibiotic. This natamycin normally occurs in the soil as a result of natural biological bacterial fermentation, and the commercially available natamycin, which is called as Natamax R, the trade name, it is a family of fungal solutions which is used in food preservation in wide variety of foods, especially against damage caused by yeast and molds, fungi. And it's functional over a wide range of pH values. It is also heat stable, which means that irrespective of the range of pH and irrespective of the temperature of processing, this is stable, this will not degrade, thereby providing the antifungal activity by preserving the food concerned. And it has also been proven to reduce yeast and mold growth in many different types of food. The third bacteriocin which we are going to talk about is Subtilin. This antibiotic was discovered and developed by scientists at the Western Regional Laboratory of USDA. It's structurally similar to Nisin, but it is produced by some strains of Bacillus subtilis. This is effective against gram-positive bacteria. It is stable to acid. It possesses enough heat resistance to withstand destruction at 121 degrees centigrade at 30 to 60 minutes. Now, this is actually very important. Synthesized by Bacillus subtilis, this is gram-positive bacteria, effective against gram-bacteria. Stable to acid, which means that the pH level has to be controlled. Second is, normally this destruction at 121 degrees centigrade for 30-60 minutes. Normally, this is what we apply for autoclave and boilers for making, especially in the lab, for killing microbes, for killing even the dormant spores. Okay, and since this temperature and pressure normally would be extremely high to destroy even many proteins, proteases and many antibiotics, because they will not be heat stable, they will not be able to withstand the temperature and pressure of this level. But Subtilin is capable of heat resistance to withstand this particular temperature and pressure, and it is effective in canned foods at levels of 5 to 20 ppm. It prevents the outgrowth of germinating endospores, and its site of action is the same as for Nisin. And like Nisin, it is used neither in the treatment of human or animal infections nor as a feed additive, and this antibiotic may be effective as Nisin, although it has received little attention till 1950s. So, even though this has been discovered by 1950s and its usefulness has been experimented and proved, the commercial application of Subtilin is not as wide as Nisin, but its potential is huge. Subtilisin enzyme preparation is derived from Bacillus subtilis, expressing the subtilisin gene from Bacillus amyloliquefaciens, and this is the one which is normally commercially used in food processing. The first part where we are discussing about the bacteriocins which are naturally used as a food preservative ends here. We will be continuing the discussion in the next part of the lecture series. Thank you.